Hello everyone, welcome to my biology classes. So from today I will be starting with a new chapter, that is your animal kingdom, chapter number 4 from the class 11 biology. So let's see why we need to classify, because we will be studying animal kingdom. So we'll see the classification, we'll study the 11 phylum. So what is the need for their classification and what is the basis of their classification, we'll see in this video. Okay, this is the introductory part of this chapter, and from next video I'll bring one phylum. Okay, so let's see that since we know we have a diverse of biodiversity, we are having different species of animals, so we need to categorize so in sequence, so that for your convenient studies. Okay, so let's see what is written here. There are over millions of species of animals, we know that have been described till now, which has been discovered till now. So these vast species has diverse structural forms, so they are having a diversity of structural form, they look different. Okay, they are having different adaptation also. Okay, so therefore the need for classification of animals become very important. So we need to classify these animals. Some are terrestrial animals, some are aquatic animals, some are having different body structure, some are having cellular structure, some are having tissue level of organization, like that. Levels of organization we are going to see, whether they are having body cavity or not, whether they are symmetrical or asymmetrical, like that we are going to study in this video. Okay, understanding? So moving on to the slide. So 11 phylums we are going to study, so you have to know their names properly. Starting from phylum number 1, that is Porifera, Coelenterata, the second one. The third one is Ctenophora, C is silent here. Then Platyhelminths, then Nematoda, then Annelida, then Arthropoda, then Mollusca, then Echinodermata, then Hemichordata, then Chordatas. Okay, so one by one I will be making a videos for each of them. All the 11 phylums I will be making. Okay, and details, characteristics, features we are going to study one by one. Okay, so moving on to the next slide, we'll study the level of organization, how the organism are classified, how the different phylums are categorized, okay, based on the levels of organization. Whether it is the first level of organization is your cellular level, okay, the second one is your tissue level, okay, the third one is your organ level, then the fourth one is your organ system level. Since we know cells are known as the basic structural and functional unit of a life, so first form the unit is the cell, then after the cell will clubbed up, all the cell will clubbed up together to form the tissues. And when the different types of tissues they used to clubbed up to form a particular group, then they form a particular organ, the organ levels. Then all the organs clubbed up together to form a particular system, that is called your organ system level. Okay, so one by one we'll go in details what it is. Okay, levels of organization is of 4 types. So moving on to the next slide, that is the first level, that is called the cellular level. Means here it is talking about cells only. The cells do not modify to form the tissues here in this case. That's why it is called the cellular level. In this level, the body shows some division of labor. It is showing some division of labor, means the cells will have different organelles, right, and different organelles will have different function. So it will therefore it is saying the division of labor means the work has been divided into different organelles. Okay, the body constituent of many cells arranged as, so it is multicellular. It is saying many cells means it is multicellular. As loose cell aggregate, they are present as loosely packed loose cell aggregate, but the cells do not form the tissue. They do not form the tissue in this case. Okay, so here the Example will be the porifera, which includes the sponges, the first phylum we have talked about. Okay, so they are having cellular level of organization. Is it clear? So moving on to the second level of organization, that is tissue level. Means in this case the cells clubbed up together to form till tissue level. Okay, then the tissue won't clubbed up together to form the organ level. Okay, so here it is saying the cell performing the same functions, the one which is performing the same function are arranged into tissue. They will clubbed up together in groups and forms the tissues, and hence they are called tissue level organization. And in which phylum you can see tissue level organization? In Coelenterata and Ctenophora you will see the tissue level of organization. Phylum 2 and phylum 3 you are going to see the tissue level of organization in Coelenterata and in Ctenophoras. Okay, okay, then moving on to the third level of organization, that is the organ level organization. Here the tissues are grouped together to form your organs. The tissue will clubbed up together to form the organs. Okay, they do not form the organ system. You can't find any kind of organ system. Each specialized for a particular function. They will have different functions. So they are the organ level organization. Okay, so where it is present? In platyhelminths it is present. Phylum number 4, that is platyhelminths, it is present the organ level. Is it clear? Okay, then moving on to the fourth level of organization, that is organ system level organization. Here the organ has associated to form functional system. The organ will clubbed up together to form a functional system, specialized system, that is your organ system. Each system concerned with a specific physiological function. They will have a specific physiological functions. Okay, it is called the organ system level of organization. So where you can see this? In annelids, arthropods, mollusca, echinodermata, chordatas, you can see the organ system level. The rest of the phylum you can see this organ system level. Is it clear? So here the cells are clubbed up together to form your tissues, and tissues will clubbed up together to form the organs, and the organs will clubbed up together to form organ system in this case. So what are the examples of it? They are Annelida, Arthropoda, Mollusca, Echinodermata and Chordatas. Okay, so is it clear to you all the four levels of organization? So let's take a quick recap. So what we got in the phylum 1, Porifera. So all the 11 phylums again I am repeating. In Porifera we have seen the cellular level of organization. Okay, and in Coelenterata and Ctenophora we have seen what? Tissue level of organization. Is it clear? And in case of platyhelminths we have seen the organ level of organization, and the rest of it we have seen the organ system level of organization. It is clear to you all now? One level we have got the cellular level in case of Porifera. Second we got the tissue level in case of Coelenterata and Ctenophoras. And third level, the organ level, we have seen in platyhelminths, and the rest of the phylum we have seen, the fourth one, that is organ system, we have seen in these phylums. Okay, so all the 11 phylums you have to know the names, okay, properly. So let us move on to the next slide. So based on the digestive system, it has been classified into two. Okay, let's just consider the digestive system. The digestive system which are having a single opening, means one particular opening will be there that will act as mouth and anus both, means it is incomplete. So where you can see in platyhelminths, you can see incomplete digestive system. Okay, and in case of since we are having two openings, we are having both mouth and anus, so we are having a complete digestive system with two openings. All right, so this is the digestive system we have. This is our mouth, so where food we used to ingest, and the anus we have where the waste material used to go out. So digestive system is of two types. One is incomplete digestive system, one is complete digestive system. Incomplete digestive system we can see in platyhelminths. Is it clear? Then moving on to the next slide, the circulatory system. Even the circulatory system means what? What do you mean by circulatory system? The heart and the blood capillaries. Okay, they are basically the circulatory system because they circulate the fluid materials. Okay, so circulatory system are of two types, that is open one and a closed one. In case of open one, the blood is pumped out of heart. The blood used to pump out of the heart. Here cells and tissues are directly bathed in it. Means the cells and the tissues used to float in the blood itself. Okay, but in case of closed circulatory system, blood is circulated through a series of vessels like arteries, veins and capillaries. They used to travel through arteries, veins and capillaries. Now can you tell me what type of circulatory system do we have? Yes, we are having a closed type of circulatory system. Our tissues and cells are not directly bathed into the blood. So we have different vessels for it, for the transportation of blood to take place. For oxygenated blood we have arteries, for deoxygenated blood we have veins. Okay, capillaries are there. Okay. Is it clear about digestive system and circulatory system? Okay, then moving on to the next slide. So let's study about symmetry. What do you mean by symmetry? Whether a particular body is symmetrical or not. Means it is in a balanced proportion or not. If we divide one particular body, suppose this is a body of some animals or a plant, we are dividing it. So we are getting two equal halves or not, or we are getting four equal halves or not, like that. Whether it is in a balanced proportion or not, whether left hand side is equal to right hand side or not, it is talking about. Is it clear? What is symmetry? The one which is having a balanced proportion. It is balanced in a particular proportion. Okay. So let's see the types of symmetry. First type is your asymmetricals, means whenever we try to divide a particular body, we can't get two equal halves. Okay, means they are asymmetricals, means they are what, like this. In case of sponges, see, the left hand side is not equal to right hand side. Just remember that. If you divide it, this portion and this portion is completely different. So in the definition, what it is saying, animal in which any plant passes through the center does not divide them into equal halves. Where any plant we are taking and we are dividing it through the center, we are not getting equal halves. We are not getting two equal halves. So in sponges are generally asymmetrical. Sponges from the phylum Porifera, the first phylum. Okay. So let's move on to the next slide. That is, it is talking about symmetrical. I don't know why this box is coming. Just ignore it. The next one is symmetrical. Here the body of some animals can be divided into two similar equal halves by one or more planes. Okay, so we can divide it to equal halves. Okay, each animals are called symmetrical animals. So this based on the symmetrical, so it is again further divided into two types of symmetries. Understanding symmetry has been divided into two types, that is asymmetrical and symmetrical. You understood what is asymmetrical and symmetrical, and this symmetrical is again further divided into two different types. That is, the first type we will talk about in the next slide, that is your radially symmetrical. Okay, you can see in the picture the starfish has been divided into many different planes and you are getting equal halves. Okay, so here it is saying, when any plan passing through the central axis of the body divides the organism into two identical halves. So any plan it is dividing into and you are getting two identical halves, then it is called your radially symmetrical. So any plan you are taking and dividing the organism and you are getting the identical halves you are getting. So where you can see starfish you got, and where you can see this radially symmetrical, you can see in Coelenterata, in Ctenophorus and Echinodermata, you can see radially symmetrical. Is it clear? Okay, then moving on to the next slide. Radial symmetry you understood, then comes to the bilateral symmetrical. Here, in some animals, body can be divided into identical left and right halves in only one plan. So it is talking about only one plan, and in case of radially symmetrical, many plans they have told in the definition, many plans. Okay, and there you are getting the equal identical halves. Here also you are getting left and right halves, you are getting equal halves when you divide it in one plan. Okay, this is called bilateral symmetrical. In case of annelida and arthropoda etc., you will get this type of bilateral symmetrical. Okay, is it clear? Symmetry is divided into two, asymmetrical and symmetrical. Asymmetrical example you got poriferous, especially the sponges. Then symmetrical, two types you got. One is radially symmetrical, which we can divide it into many plans, and bilaterally symmetrical, we can divide it into one plan, and we are in both of them we are getting identical halves we are getting. Is it clear? And in case of radial symmetrical, we got the example of Coelenterata, then Ctenophorus, okay, and Echinodermata we got. And in bilaterally symmetrical, we are getting Annelida, arthropodas, etc. Is it clear? Symmetry is clear to you all? So let's have a quick revision from this again. We are bringing the 11 phylums. Okay, 11 phylums. So porifera we got, they are asymmetrical. Coelenterata, Ctenophorus and Echinodermatas are what? Radially symmetrical. Okay, and the rest of them are your bilaterally symmetrical. Is it clear? Porifera, asymmetrical. Coelenterata, Ctenophora and Echinodermata, radially symmetrical, and the rest of them are bilaterally symmetrical. Clear? Okay, then moving on to the next slide. Let's talk about germ layers. What are these germ layers? So let's talk about it. These are groups of cells behaving as a unit during early stage of embryonic development. So they are a group of cells which behave as a one single unit during the embryonic development. Okay, it differentiated to give rise to all the tissues, organs of the fully formed individual. So this germ layer will give rise to your tissues and organs in the later stage. Okay, so what happened during embryonic development? Suppose a single cell stage you are getting, then 2 cell stage you will get, then 4, like that. Equal division you can see, 4 cell stage. Okay, so what happened whenever after crossing this stage, they will have some layering and they will act as a single unit. Okay, that layering is called your germ layer. Is it clear? And this layering will give rise to the different layers it will have. The germ layers are of 3 different types. In some they are having 2 different types. So this will give rise to your different tissues and different organs, okay, of that particular individual. Is it clear? So moving on to the next slide. On the basis of germ layer, animals can be classified as follows. The first one is the diploblastic animals based on the germ layer. Diplo means 2. Diploblastic animals means it will have 2 germ layers. It will have 2 germ layers. Okay, so they are having 2 embryonic layers. These germ layers are also known as your embryonic layer. Okay, so external, the outer one, ectoderm is present. The outer one, ectoderm is present, and in internal endoderm will be present. EN I am writing. It will have 2 germ layers, the outer ectoderm and the inner endoderm. Okay, in addition, an undifferentiated layer, one more undifferentiated layer in the middle, which is not considered as a layer. Okay, it is undifferentiated since, so that is known as mesoglea, is present between the ectoderm and the endoderm. Okay, so where you can see this diploblastic organism, you can see in case of Coelenterators and Ctenophorus, you can see the diploblastic layers. Okay, 2 different layers you can see in case of Coelenterata and Ctenophorus. Is it clear? Okay, then moving on to the next slide, the triploblastic animals. Okay, triplo means 3, so it will have 3 different germ layers. Okay, it will have outer same, the 3 different germ layers, the ectoderm, endoderm, and they will have a middle layer called mesoderm. So mesoderm will be there, outer ectoderm will be there, inner endoderm will be there. Okay, 3 different layers will be there. So where you can see this triploblast layer, you can see this in phylum Platyhelminths to phylum Chordatus. From Platyhelminths till Chordatus, you can see triploblastic layers. Is it clear? You won't see any germ layer in case of Porifera. It starts with Coelenteratas. Then in Coelenteratas and in Ctenophorus you will get the diploblastic layers, and rest of them you are getting the triploblastic layers. Okay, just remember ectoderm, endoderm and mesoderm. Okay, the 3 germ layers. So moving on to the next slide, again I am bringing the 11 phylum. So Porifera does not have any kind of germ layers. Okay, Coelenterata and Ctenophorus are diploblastic. Okay, and the rest from Platyhelminths to Chordatus, they are triploblastic, means they are having your 3 germ layers. They are having 2 germ layers, ectoderm and endoderm they will have in Coelenterata and Ctenophorus, and in case of from Platyhelminths to Chordata, you will have ectoderm, mesoderm and endoderm. Is it clear? And Porifera does not have any kind of germ layers. Okay, germ layer, or you can say embryonic layer also. Is it clear? Then moving on to the next slide. So this is the picture showing diploblastic germ layer, that is mesoglea, undifferentiated layer in the middle, ectoderm the outer one, endoderm the inner one. Okay, this is triploblastic, where you can see the middle mesoderm, this is middle mesoderm, the outer ectoderm and the inner endoderm. Is it clear? This is the diagram beautifully showing the diploblastic and triploblastic germ layers. Okay, then moving on to the next slide, let's talk about coelom. Coelom means what? It is body cavity. It's a body cavity. Okay, a body cavity that forms from the tissue derived from mesoderm. So we have studied already what is mesoderm. Mesoderm is a part of germ layer which is between the ectoderm and the endoderm. From this mesoderm there will be there will be a formation of body cavity that is called your coelom. Okay, so now we will study the three different types of coelom which are present in different organisms. Okay, so let's start with the three different types. This is number one type. The presence of body cavity that is lined by mesoderm means the mesoderm used to modify to form the body cavity, okay, that is lined by mesoderm, that is called your coelomate organism. Okay, they are called as coelom. Okay, and the presence of patches of mesoderm in the body cavity. So whenever in the body cavity there are patches of mesoderm, here the mesoderm is completely lining the body cavity. Okay, if there is a body cavity, the mesoderm is completely lining the body cavity, then it is coelomate organism. Coelom means. Then the second one, it is saying when the presence of patches. So in the body cavity there are patches. There are patches of mesoderm. We'll see the figure in the next slide. Patches of mesoderm is present in the body cavity, then it is called pseudocoelomate. False coelom means pseudocoelomate. And whenever there is a total absence of body cavity is called the acoelomate. When there is no such body cavity, then it is called acoelomate. Getting my point? This body cavity has been divided into three different types, that is coelomate, pseudocoelomate and acoelomate. In case of coelomate, the body cavity is lined throughout by the mesoderm. Here the body cavity is not lined throughout by the mesoderm. It is having some small mesodermal patches, but here in this case in acoelomate the body cavity is completely absent. Is it clear? Then moving on to the next slide. So examples where you can see coelomate, we can see in case of Annelida, Mollusca, Arthropoda, Echinodermata, Hemichordata and Chordata, the last phylums which are present actually. Okay. Then pseudocoelomate, the false coelom, which where it is present in case of Acyhelminths, we can see pseudocoelomate. Coelomates which are not having any kind of body cavity, we can see in Platyhelminths. Is it clear? In Platyhelminths we can see. Okay. In case of this Coelenterata and Ctenophorus, there is no such coelom concept because there is no mesoderm. Okay, and in case of Porifera there is no germ layer only, so there is no question of any coelom. Is it clear? So this coelom concept starts from Platyhelminths. Is it clear? Then moving on to the next slide. So this is a picture showing the coelomate, pseudocoelomate and acoelomate. So here coelom is present which is lined by a mesodermal layer. Here pseudocoelomate where you can see a body cavity having patches of mesoderm, and here the body cavity is absent. Is it clear? Then moving on to the next slide. So here you can, again I have given the 11 phylums. Okay. So you can easily identify. This one won't have any relation with the coelom because here this mesoderm is absent. Here germ layer is only absent. In case of Porifera, germ layer is only absent. In case of Coelenterata and Ctenophorus, here it is diploblastic since here is no mesoderm, so no question of any coelom. Then Platyhelminths, from Platyhelminths it will start. It is acoelomate. Then comes what? it is pseudocoelomate, then this will have, they are coelomate. Is it clear? Then moving on to the next slide, let's study about a new topic that is called your segmentation. You know what is segmentation? Okay, so what it is saying here, in some animals the body is externally and internally divided into segments with a serial repetition segmentation. Okay, so here the body is divided into segments, so they are having segments like this. Internally also they are having segments, externally also they are segments. Okay, and they are repeated sequence of segments parallelly arranged. Okay, so they are called as segmentation, and based on this it is of two types. Okay, segmentation is of two types. One is metameric segmentation. This is very important, metameric segmentation. You can see this in case of earthworm. So you can see in this picture a picture showing earthworm, which is showing your metameric segmentation. They are segments. Okay, so what it is saying, a segmentation that simultaneously divides body both externally and internally. The body has been divided into, that is called metameres or metameric segmentation. This kind of segmentation you can see in case of annelids, arthropods and chordates. You can see this type of segmentation. Both internally and externally it is divided. Is it clear? Metameric segmentation. Okay, then moving on to the next slide. Now it is saying pseudometamerism, the next one, the second one. Segmentation has divided into two types, that is metameric segmentation and pseudometamerism. Okay, means false metamerism it is. Okay, where you can see this false metamerism, you can see this in case of tapeworms. This is a diagram showing a tapeworm. Okay, here the body is divided into parts or segments called proglottids. So here also the body has been divided. See, it is divided into segments, and these segments are here, it is called proglottids. Is it clear? They develop from the neck region. So from the neck region it will start dividing. Is it clear? But are not embryonic in origin. They are not embryonic in origin. In case of metameric segmentation, from mouth till anus you can see segmentation. Is it clear? In pseudometamerism you can see segmentation from the neck region onwards. Is it clear? And metameric segmentation example you can give for earthworm. So you can see metameric segmentation in case of earthworm, and pseudometamerism you can see in case of your tapeworms. Is it clear? Okay then. Is it clear? Segmentation topic is done. So next, the last topic is your notochords. Okay, this is the last topic from this video. So what it is? It is a mesodermally derived rod-like structure formed on dorsal side during embryonic development in some animals. So what it is saying, it is mesodermally derived. So three germ layers we have studied. Ectoderm, mesoderm and endoderm. Okay, so it is mesodermally derived. Okay, coelom is also mesodermally derived. Okay, and notochords are also mesodermally derived. They are rod-like. How the rods are, the structure of rod, like that it is the shape. Okay, form on the dorsal. What do you mean by dorsal side? Dorsal means back side. Okay, ventral, one more term is there, ventral. Ventral means your front side. Dorsal is your back side. So which are present in the back side of the body. Okay, so during embryonic development you can see this notochords. Is it clear? And the animals which are having this notochords are called the chordates, and the animals which do not have any kinds of notochords, they are called non-chordates. Means all the phylums from phylum number 1 to phylum number 10, then from Porifera to Echinodermatous, so they are what? They are non-chordates. Okay, the last phylum is your chordates, means they are having the notochords. Is it clear? It is a rod-like, it is mesodermally origin, it is present in the dorsal side of the animal. Okay, and the animals which are having the notochords are called your chordates, and the animals which do not have any kind of notochords are called non-chordates. Clear? So let's see a picture of a notochord. See, this orange color, this one, this is your notochord, which is rod-like also, present dorsally also. Is it clear? This is your notochord, which are present in the chordates. Is it clear? So that's all for today's video. This was the introductory part from the chapter The Animal Kingdom. The next subsequent video I will be bringing on different phylum. The total 11 phylums I'll bring in one videos I'll talk about. Okay, so thank you for your patient hearing.